Hi there, my name is Dr. George Murphy, and I'm one of the founding co-directors of the Boston University and Boston Medical Center's Center for Regenerative Medicine, which we affectionately refer to as the CReM. In a nutshell, we're a group of like-minded researchers interested in all things stem cell biology and regenerative medicine related. David Seldin, who's being honored today by this symposium, was my division chief, and I've always felt really special because he was also the guy who recruited me to Boston University, thereby initiating my independent research career. David was my mentor, who was always there for me, intellectually, emotionally, spiritually, and pretty much any other way you could possibly think of. He was the kind of guy who would see my trainees leaving late at night, and when he would find out they were heading to the pub to have a drink, he'd be the guy that would drop them off. David was my friend and my biggest fan. He was always amazingly supportive of the CReM, and from the very beginning was one of our biggest patrons. At the CReM, one of our main directives is to focus on diseases which directly affect the underserved community at Boston Medical Center. And this simply echoes David's personal philosophy of giving rather than taking in this world. I'm sorry that I cannot be with you today in person, but I like to think that where I currently am and what I'm currently doing also honors David and fulfills some of the dream translational applications of our work that we would talk about late in the evening in his office. So where am I exactly, you ask? On March 18th, I traveled from my home base at the Center for Regenerative Medicine at 670 Albany Street in Boston overseas to Japan, and more specifically to the Kyoto University Hospital, where we are currently initiating the first-in-man trials of stem cell-derived blood products. So how will this work exactly? Well, one of the cornerstones of what we do at the CReM is the use of induced pluripotent stem cells, or iPSCs, which have the ability to become any cell or tissue type in the body and are an exact genetic match to the individual from whom they are created. So with this in mind, the hard part is figuring out how to guide these iPSCs to blood cells using a process we refer to as directed differentiation. Nature, i.e. the human body, is exceptionally adept at this process, and each one of us makes an amazing 2.5 million red blood cells every second of every day, and more platelets than there are stars in our Milky Way galaxy. Historically, it's been very challenging to develop an in vitro platform that could generate clinically relevant numbers of blood cells. So our approach mimics the natural sequence of blood cell development. Our methodology is short, completely chemically defined, and requires no feeder cells or serum, thereby making good manufacturing practice production and clinical translation feasible. Using this protocol, we can create large numbers of hemoglobinized iPSC-derived red blood cells, as well as polyploid megakaryocytes, which have the ability to produce functional platelets in culture, as noted here in the following movie in which an iPSC-derived megakaryocyte is seen producing proplatelets. Putting this all together, in Japan, harnessing technology developed right here at Boston University, we are using the directed differentiation of iPS cells into megakaryocytes that produce functional platelets, which will then be infused into patients with chemotherapy-induced thrombocytopenia. In this way, we hope to be able to obviate the need for blood cell donation and circumvent issues related to contamination or supply. In addition, here at BU, with the support of the CTSI and in collaboration with Dr. Marty Steinberg and the Center for Excellence in Sickle Cell Disease headed by Liz Klings, we are also in the beginning stages of initiating a trial in which patient-specific iPSC-derived erythroblasts will be infused into patients suffering from sickle cell disease to meet their transfusion demands and potentially ameliorate some of the complications of the disorder. So this is where our story ends, but I prefer to think of it as the beginning, as the work we all do honors David's life and carries on his legacy. With that in mind, in essence, we are all David Seldin. Seldin, our patron, mentor, friend, and inspiration.
We are the CReM!